What is kinesin and dynein? A kinesin is a protein belonging to a class of motor proteins found in eukaryotic cells. Kinesins move along microtubule filaments, and it is powered by the hydrolysis of adenosine triphosphate. Thus, kinesin is an ATPase, a type of enzyme. The active movement of kinesin supports several cellular functions including mitosis, meiosis and transport of cellular cargo, such as in axonal transport and intraflagellar transport. Most kinesins walk towards the plus end of a microtubule, which in most cells entails transporting cargo such as protein and membrane components from the center of the cell towards the periphery. This form of transport is known as anterograde transport. In contrast, dyneins are motor proteins that move toward the minus end of a microtubule in retrograde transport. Clostridium tetani toxin, poliovirus, rabies virus, and herpes simplex virus use dynein for retrograde transport to the neuronal cell body. Herpes simplex virus reactivation occurs via anterograde transport from cell body kinesin-mediated. Slow anterograde transport is the rate-limiting step of peripheral nerve regeneration after injury. In an experiment, laboratory animals are subjected to a toxic insult that specifically targets the protein kinesin. Which of the following is most likely to be absent from tissues on histological examination as a result? The correct answer is B, secretory vesicles in nerve terminals. Kinesin is a microtubule-associated motor protein whose function is anterograde transport of intracellular vesicles and organelles toward the plus, rapidly growing, ends of microtubules. Kinesin uses energy derived from ATP hydrolysis to move along the microtubule. The plus ends of microtubules typically lie at sites distant from the nucleus. Thus, in neurons, kinesin carries vesicles and organelles away from the cell body and toward the nerve terminal. A targeted toxic insult to this protein would be expected to impair anterograde transport, resulting in a deficiency of synaptic vesicles at the nerve terminal. Choice A, the Nissl substance in neurons, corresponds to the rough endoplasmic reticulum. The functions of Nissl substance are thought to be the same as those of the rough endoplasmic reticulum in general, primarily the synthesis and segregation of proteins. Choice C, the transverse tubule system is a tubular network located at the junction of the A and I bands of striated myocytes. T-tubules are open to the extracellular space and facilitate the spread of depolarization to the inside of the cell. Choice D, desmosomes are protein aggregates that mediate cell-cell adhesion between epithelial cells. Desmosomes are one of the stronger cell-to-cell adhesion types and are found in tissue that experience intense mechanical stress, such as cardiac muscle tissue, bladder tissue, gastrointestinal mucosa, and epithelia. Choice E, microvilli are small cylindrical extensions of the apical plasma membrane that contain actin-thin filaments. Microvilli cover the apical surfaces of intestinal epithelial cells. They do not contain microtubules or microtubule-associated proteins. Educational objective. Kinesin is a microtubule-associated, ATP-powered motor protein that facilitates the anterograde transport of neurotransmitter-containing secretory vesicles, down axons to synaptic terminals.